وعليكم السلام دكتور.
Okay, we'll continue with yesterday's class. Before we continue, yesterday one of your friend asked that what is whether the rhesus factor is beneficial or not. So when I was talking about antibodies, maternal antibodies, prepared antibodies from the mother to the fetus, that is the advantage. Okay, because they are already prepared antibodies manufactured within the mother's womb, and it is sent to the fetus so that the fetus can fight some of the infections and other things. But the rhesus factor antibody, this is different from this one. Okay, so these are the ready-made antibodies, and this is the antibody produced because of the mixing of blood between the fetus and the mother during the first delivery. Okay, when the mother is Rh negative and the child is Rh positive. So this is disadvantage because it will create problem for the future pregnancies. So this antibody is different from these antibodies. Okay, so the maternal antibodies for all other kind of infections and other things which are produced, those are advantages. But Rh factor antibody, this is not a advantage for this. Okay, moving further, we were discussing about the placenta, and we especially I wanted to I showed you the development of the villi. If you see here, this is the syncytiotrophoblasts, and they start producing finger-like projections into the endometrium or decidua, and they start forming spaces called as lacunae. These are filled with blood now, and this is called as uteroplacental circulation. And if you see the changes within the syncytiotrophoblasts, where the finger-like projection, these villi are being formed. If you take a section of this, then we see there is changes in the initial stages. There will be only two layers. One is the syncytiotrophoblasts and the cytotrophoblast. If you take a section at this level, and I showed you that there will be, if you take a cross section, it will be only the syncytiotrophoblasts and the cytotrophoblast inside. Then later, if you see, the mesoderm starts entering inside. Okay, so now there are three layers: syncytiotrophoblasts outside, then cytotrophoblast, and inside is the extra embryonic mesoderm. Now there are three layers. If you take a section at this level, these finger-like projections now will have three layers: outer syncytiotrophoblasts, then cytotrophoblast, and here is the extra embryonic mesoderm. Then the blood vessel starts invaginating into these villi-like projections. Okay, then if you take a section, then you can see the blood vessels also. Okay, now this is called as the tertiary villi. This is primary, this is secondary, and tertiary, or it is also called as first degree, second degree, and third degree villi, or it is better to call them as primary, secondary, or tertiary. Then we discussed there are two types of villi. So this villi are depending on development. They are primary, secondary, and tertiary. These are the final stage of maturation of the villi. Okay, so these are the types depending on the development. Now here, depending on whether they are attached to both the surfaces. This is the decidual plate, and here is the decidual plate, and here is the chorionic plate. And if they are attached to both the plates, then this is called as the anchoring villi. Anchoring means attached. Okay. Then there are some free floating villi which are flowing and mainly acting in the exchange of material. So these are called as free villi. So they are anchoring villi and free villi. So if you see here, so all around this free villi will be the blood of the maternal blood, which will be there outside, and inside here will be blood vessels. Later I will show you where the fetal blood will be passing through. So there is no exchange, there is no actual mixing of the blood between the mother and the fetus. Now coming to the divisions of villi. So within the villi, what are the parts? What are the parts of the villi? Now you will see that it has three parts. That is called as the truncus cori. Truncus cori. Truncus means just like a trunk. It looks like a tree. So it is called as the trunk of the trunk, means the main part of the tree. That is called as the trunk. So this is called as truncus cori, and the branches, these are called as the ramus cori. Ramus cori, the main branches. And the smaller finer branches, these will be called as the ramuli cori. Ramuli cori. These are smaller branches. These are called as the ramuli cori. So main stem is called as this truncus cori. Then its main branches are called as the ramus cori, and the smaller branches, all these are called as the ramuli cori. So depending on the size of the villi, the parts of the villi, so they are divided into three parts: truncus cori, which is the stem; ramus cori, which are the main branches; and ramuli cori are the finer or smaller branches. Then I have already told about the chorionic villi. Chorionic villi, which are around the Destia basalis, they will survive as the chorionic fundosum, and they form the placenta, as well as the Destia basalis, which is there. Both together, Destia basalis as well as the chorionic fundosum will form the placenta. And the remaining villi, which are all around before, now they start disappearing and they start leaving. So that's why these are called as the chorionic leaf. Opposite the Deciduea capsularis, here is the Deciduea capsularis, and this is the Deciduea parietalis, and the basalis is here. Okay, so the near the Deciduea capsularis, all these villi start disappearing. So this is called as the chorionic leaf. Okay, now coming to some more parts of the structure of the placenta. So by the beginning of the fourth month, the placenta has two components. Okay, one is the fetal portion, which I have shown you, the chorionic fundosum. As I showed you, this is the chorionic fundosum, and the maternal part, that is the Deciduea basalis. Here will be the Deciduea basalis. Okay, so the fetal part, that is called as the chorionic fundosum, and the maternal part called as the Deciduea basalis. And the chorionic and decidual plates I have already shown. Here this will be the toward the fetus, that will be called as the chorionic plate, and the other side toward the decidua, that will be called as the decidual plate. Borders placenta on fetal and maternal side respectively. Okay, so between these two plates, one is the chorionic plate, and the decidual plate is the whole of the placenta which is being formed. Junction zone is between two plates and contain intervillous space. If you see here, these are the villi. Now you can see here there are small villi, there are big villi. Okay, and in between you can see space. This is called as the intervillous space, which was initially lacunae. Now it will be filled with blood here. So this is called as the intervillous. Between inter means between villi. That's why it's called an intervillous space. Decidual septa that projects into the intervillous space but not reach the chorionic plate. If you see these anchoring villi start from the chorionic plate and goes up to the decidual plate. Okay, and they are called as the anchoring villi. But similarly, there will be some septa formed from the decidua, but they don't reach up to here. Okay, so they will divide the whole of the placenta into smaller lobes. Okay, but they are incomplete lobes. Okay, these are called as the decidual septa that projects into the intervillous space. This is the intervillous space, but not reach the chorionic plate. It doesn't go up to the chorionic plate here. This is the chorionic plate. Okay, so they will be dividing, and you can see it as cotyledons. The space between the two septa will be called as the cotyledons. Cotyledons, the placenta is divided by decidual septum into cotyledon compartments. So the space between the two septa is called as the one cotyledon. So as we can see, there are multiple cotyledons here. Okay, which is seen from the maternal side, because from the fetal side it will be very smooth, covered by amnion. Okay, and we can see, how do you identify? By the presence of the umbilical cord. Umbilical cord will be present toward the fetal side, not the maternal side. Maternal side, you can see the cotyledons, what you are seeing here. Now coming to the circulation of the placenta, how there will be circulation taking place. If you see, the intervillous space of the mature placenta contains approximately, usually almost 150 ml of blood coming from almost 80 to 100 spiral arteries. Let me show you. Okay, so here you can see, the spiral arteries are within the endometrium of the uterus. So now they start opening into the intervillous space. So almost 80 to 100 spiral arteries opening into this intervillous space will give a blood quantity of almost 150 ml of blood always present in the placenta for the circulation between the fetus and the mother, which will exchange about 3 to 4 times per minute. So this 150 ml of blood is not only staying there, it will be recirculated, so that whenever there is exchange of gases has taken place as well as nutrition, it will be removed out and the new fresh blood will come. So it will almost 3 to 4 times per minute this 150 ml of blood is change. Okay, so that new blood can come and there can be better exchange. The fetal blood arrives to the placenta by two umbilical arteries. As we already seen, there are two umbilical arteries shown in the, which actually should be in the blue color. Okay, that branches to form the capillaries in the villi, where the gaseous exchange occurs with the maternal blood in the intervillous space. So these, if you see here, these are the two arteries. So they are bringing the deoxygenated blood to get the pure oxygen from the maternal blood will be flowing here. So these are the villi. So you can see here the blood is coming from these are the arteries which are shown in blue color. So I said this is unique, that arteries are not shown in red but they will be shown in blue because they'll be carrying the deoxygenated blood or the CO2. Okay, and it will be exchanged here in the intervillous space. So here will be the maternal blood filled. Okay, so they will give the oxygenated blood and it will pass through back into to the fetus through the umbilical vein. So pure blood is coming to the fetus through the umbilical vein. Okay, so fetal blood arrives to the placenta by two umbilical arteries that branch to form the capillaries. As you can see here, these are smaller branches called as capillaries. Okay, in the villi where the gaseous exchange will be occur. So you can see the same thing. So this is the maternal arteries coming here, and the blood is here in the intervillous space. So there will be exchange of gases taking place. Finally, the oxygenated blood returns to the fetus via the umbilical vein. So the oxygenated blood comes through the umbilical vein. Okay, so umbilical. So that's why it is shown in red. So there is a wrong representation here in the colors. Actually, this this is the vein. It should be shown in red, not in blue. Okay. Okay. Coming to the placental
Doctor,
yeah,
sorry, but your voice is not totally clear. There is
not
a noise.
there is a noise. Okay.
Yeah, like someone move something.
Okay. Let me put the microphone. Okay, now so this will be the exchange of gases taking place here. Okay, so as we have already talked about the placental barrier or the membrane, maternal blood in the intervillous space and the fetal blood in the capillary don't mix, as I said. So there is a space or there is a barrier between the maternal blood and the fetal blood. Okay, so they are separated by these four layers which are called as the placental barrier, which we already discussed. Starting from outside to inside, there is syncytiotrophoblasts, cytotrophoblast, extraembryonic mesoderm, and the endothelium of the fetal capillaries. Okay, so there will be four layers. But by the fourth month, the second and the third layer disappear. These two layers disappear. So if you see here, so initially there will be four layers. So outer will be the syncytiotrophla. If you take a section of this one, this is the section of this one. Okay, one of the villi. So here outside we have the syncytiotrophloblast. Here this is the cytotrophoblast. Here is the extraembryonic mesoderm, and here will be the blood vessels which are lined by the endothelium. So blood outside here will be maternal blood, and here will be the fetal blood. Now for exchange of gases, it has to cross the four layers. But by the fourth month, these blood vessels will come closer to the wall and it becomes just two layers, the first and the fourth layer. So you can see the syncytiotrophoblasts and the endothelium. The connective tissue and the cytotrophoblast have almost disappeared. So by the fourth month, the barrier becomes much more thinner. If you see here, this is again shown here. So there are four layers. Okay, four layers here. So the blood is coming here into an intervillous space, and here are the villi, and there will be exchange of gases. If you take a section of this, there are four layers, but by the fourth month and so afterwards, or it will become just 2 layers, so it becomes very thin. Okay, so there are chances of crossing of much more materials by this time. So you should be extra careful after the fourth one, because the membrane becomes much more thinner. Okay, so
Sure.
this is all about this lecture which we discuss. Now we'll go to the second lecture of today's lecture, which
Lecture.